To one and all present over here. Uh, Faculty of Agriculture, College of Agriculture are always organizing different interactions with the eminent personalities in the sector of agriculture and allied sciences. Friends, as you all know that agriculture is one of the most important and emerging field in the human civilization. And due to advancement of the science, we have different branches come over in the recent years. And agriculture, among agriculture and allied sciences, bioprospecting is the one thing that we are going to have interaction by our today's chief guest, Dr. Bharin, Institute of Science, Bangalore Institute. And as all of us know that the bioprospecting of plants and living organisms for pharmaceutical purposes is useful not only to the pharmaceutical firms but also to the host country and the local people who are benefited from the ownership of biological research. So in this regard, today we have with us Dr. Madhuri Kumari from IIS Bangalore, one of the premier institute of the India in the science sector. And for the audience, she is the student of our respected dean sir, and she has before that, I would like to thank Honorable Dean sir for providing me opportunity to moderate this introduction. I would like to request our chief guest, Dr. Madhuri ji, to deliver her speech and enlighten our ideas about bioprospecting in relation to agriculture or healthcare. Please, ma'am.
Yeah. Thank you, sir, for the introduction, for the kind introduction. So, yeah, I will be good afternoon, everyone. And so today I'll be dealing with fungal bioprospecting and their utilization from agriculture to healthcare sectors. And before I begin, I give my brief introduction. I am Madhuri, and I am currently working in Department of Biochemistry, Indian Institute of Science, as CSIR pool scientist. So, just a minute. Yeah. So, what is bioprospecting? Bioprospecting is also known as biodiversity prospecting. So, as Sir told, that bioprospecting has been used widely from plant origin, from animal origin, and from multiple sources. And the bioprospecting is the thing that we can use. We can use the microbes, the animals, the plants for the benefit of human and mankind without hampering the natural process, without hampering the nature. So, if we go by the explanation, biodiversity prospecting is exploration of biodiversity for new biological resources of social and economical values. And the bioprospecting, as we can see, it is associated with mankind from since ages. Since ages, our ancestors, the tribal persons, the indigenous population is using the traditional knowledge so that they could cure the diseases, they could synthesize the dye, and they could also use the traditional knowledge. You can use the potential knowledge of nature in for their development. What kind of things that can be isolated or can be utilized in case of bioprospecting? So there are multiple of things which can be isolated or utilized from the natural sources, such as enzymes, such as drugs, macromolecules, dyes, biochemicals, and genetic information. Say if I say cellulase, chitinase, these are the enzymes which are isolated from multiple of microbes. Drugs, as we can see, one of the most potential drug isolated from plant is Taxol, which is widely used against cancer. And till 2014, almost 1/3 of the drug isolated, as per the FDA, was isolated from natural sources through bioprospecting. Multiple of dyes have been obtained from plants, from animal sources, from corals, and genetic information, genetic engineering is a new aspect which has been added in the bioprospecting aspect. So, bioprospecting has been used in multiple aspects of human life, such as medicine, pharmaceutical science, agriculture, aquaculture, bioremediation, nanotechnology, cosmetics. These are few. Apart from that, multiple of fields are being touched by bioprospecting, and the bioprospecting has been used for the development, day-to-day development of the human society. And it plays a very important role in circular economy. In circular economy, generally 3 R's have been focused. These are reduce, recycle, and reuse. As these are obtained from the natural resources, they can reduce the pollution, and they are in sync with the natural processes. And what are the source of bioprospecting? These are plant, animal, as well as microbes. So if these are the sources available, why one should go for the microbial bioprospecting? So yeah, so microbes are quite tiny creatures, but they are quite valuable. We cannot grow the trees as fast as we can grow the microbes. So if we talk about the microbes, generally the microbes which have been used in bioprospecting are bacteria. We can see the use in PCR in our genetic lab. We use the Taq polymerase that has been isolated from Thermus aquaticus, that is a bacteria. In case of fungi, we see penicillin that is isolated from Penicillium. Actinomycetes are also a big source of production of drugs, such as we can say streptomyces. It is producing streptomycin. Many of the potent cancerous drugs are also produced by actinomycetes. And cyanobacteria, they are used in production of biodiesel, biopetrol, bioplastics, and many more. Many of the industries are turning towards the biogases and bio-based petrol and diesels. And viruses, you must be wondering how viruses can help us. So you see, in case of corona time, the vaccine which we have got, that is having adenovirus as a virus which is not harmful to us, but that is carrying the particular active ingredient of the vaccine. And apart from that, bacteriophages are the that kind of viruses which can kill the bacteria, and they are commercially used as antimicrobial agent. So, okay, we go to the microbial bioprospecting, but then why we should go for the fungal bioprospecting? So there are multiple of benefits associated with fungi which are not present with other kind of microbes. Say the fungal population is quite diverse. I will be dealing with all these in my next slide. So I am just going to tell how fungal bioprospecting is more helpful. The diversity of fungus is quite vast. They can be extracted from extreme locations. They can be extremophiles. They can tolerate the low temperature, low pressure. They can be produced in high amount. The culture optimization is easy, and they are prolific producers of secondary metabolites and extracellular enzymes, as well as they can reside inside the cells of some other organisms. So I'll be dealing with all of these in my next slides. So yeah, next is fungal diversity, and where we can find the fungi. Fungi can be found in any of the location. These are the trichoderma which have been isolated from soil of pathogen contaminated soil. These are the mushrooms which are quite prevalent in jungles as well as in forest. These are multiple kinds of fungi which have been isolated from different sources. These are the lichens. Lichens are also a synergistic relationship between fungi as well as algae, and they are found in multiple kind of jungles, multiple kind of places. And here you can see these are the fungi which have been isolated from inside of the green algae. These are known as endophytes, and you can see the dendrogram shows how diverse they are. That means fungal diversity is very huge, and they can be found from inside us to outside us. From any location you name them, and you can find the fungi from that. And fungi are the prolific producer of secondary metabolites as well as extracellular enzymes. So here we can see what are the secondary metabolites. Secondary metabolites are those kind of chemicals which the plant or the animals or the microbes, they don't use that kind of chemicals for their particular growth and metabolism, but they can be used to tolerate the stress. They can be used to cope up the biotic stress, abiotic stress, as well as for the removal of predation and secondary kind of things. So here we can see there are multiple kind of natural products. There are multiple kind of chemicals which can be isolated from the fungi. We can see the kind of examples are polyketides, then terpenes, then polyphenols, then peptides, then again proteogenic and non-proteogenic amino acids. There are a number of secondary metabolites, different kind of sources, phenols, polyphenols. They can be produced from different kind of fungi. And here I am showing you one of my data where from the fungi, the two kind of acids, chrysine as well as azelaic acid, has been isolated, and they can be used in multiple aspects. And this has been done by GCMS, gas chromatography mass spectroscopy. Next comes the fungal endophytes. Now, what are fungal endophytes? As I told, endophytes are something which reside inside the living organisms. So you can think, if you are if the endophytes are residing inside the living organism, they must have some special characteristic. They are having co-evolution with the host. With the host only they are growing. So they are quite diverse. They have a lot of secondary metabolites, and they may possess some of the secondary metabolites which were possessed by only their host where they are living. And they may have combinatorial biosynthesis pathway. That means they have some kind of pathway which is found either in host or the fungi, or maybe the what we can say, the synergistic pathway between them. And one important thing which endophytes have is horizontal gene transfer. That means they can have some kind of compounds which their host is having. Say if a plant, say a taxus baccata, that is producing taxol, so the plant is producing taxol. But if we isolate the fungi from that tree, there is a possibility that fungi can also produce taxol, which will be quite beneficial, because cutting a tree is like a challenge nowadays. It may hamper the environment and increase the pollution and can endanger the species as well. So endophytes can emerge as a sustainable and can emerge as a substitute for the medicinal plants. So see in this picture, there are actually more than 50 ways where we can bioprospect the fungi. But here you can see, the diagram is showing the 50 ways through which we can bioprospect the fungi. Here the biodiversity is coming, we are sequencing, and now it is used for the production of novel compound that can be used in biocontrol, biofertilizers, that can also be used in post-harvest control of diseases, as well as to stop the contamination mycotoxin produced by the fungi. And once we come here you can see the macrofungi can be used directly. They can be used in farming foods. They can be used for synthesis of multiple products, and you name n number of things, and fungi can be used. They can be bioprospected. And yeah, there are this figure shows the 50 ways through which fungus can be bioprospected. Now, the use of fungal bioprospecting in agriculture. So, how fungus can help in agriculture? So the plant basically plants have to face two kinds of stresses: biotic stress and abiotic stress. Biotic stress, that means the infection of something, that may be bacterial infection, fungal infection, insect infection, or herbivory. So, and apart from that, plants have to face multiple abiotic stresses as well. Say they may have high pH, they may have to survive in high pH and high temperature, high salinity. There are multiple of things where plant have to grow and cope up the natural conditions, where fungus can help the plants to grow. And how they do so? They help the plant in multiple processes, such as they can help in synthesis, nitrogen fixation, solubilization of phosphate, production of phytohormones. They can help the plant to compete with other pathogenic fungi, production of several bioactive metabolites, as well as inducing of plant resistance. The many fungal species which are known for their plant growth promoting activities, such as trichoderma, they induce the plant disease. They can induce the systemic acquired resistance as well as induce systemic resistance in the plant, so that plant itself can cope the different kind of stresses. So there are some mechanisms through which plant growth promoting fungi have been used in agriculture. The first figure shows the phosphate solubilization. Here we can see this is NBRI BPV media, and the decolorization show that the fungi is responsible for the phosphate solubilization. Generally, most of the India soil, say if we say Gujarat soils, also in UP also, the plants have to face the condition of sodic soils. And what does the fungi do? It decreases the pH of soil, and thus makes phosphate available to the plants. Otherwise, it is not available to the plants. And further, here you can see The fungi are also helping for production of auxin. Auxin, yes, you must be knowing this is the plant growth promoting hormone, and the fungi are inducing the synthesis of auxin. Here, plant test has been performed on maize with different kind of fungi, and you can see this is the control one, which is not treated by any of the plant growth promoting fungi. And after treatment of plant growth promoting fungi in both the conditions, either without phosphate or with phosphate, the growth, the dry weight, the height, everything is increasing. So there are multiple ways, there are multiple mechanisms through which they are doing so. And here I am showing the one of the mechanism where they increase the synthesis of chlorophyll. The application of plant growth promoting fungi such as trichoderma can increase the synthesis of chlorophyll inside the plant, and thus can increase their height, their weight, because the photosynthesis efficiency ultimately increases. And here we come. Yeah. So this slide shows that how in nutrient deficiency condition, once we apply any biofertilizer, here Trichoderma rhizae is applied as biofertilizer. And here what we are showing, there is one rice variety known as Hina, and that is drought resistant. And here we are supplementing only 50% of nitrogen. And this is the control one where the plant is deficient in nutrition. And in this, you can see that plant has been supplemented with biofertilizers. And there is a clear difference between the anatomy of the plant. You can see the differences here, that parenchyma, the population of metaxylem has been increased. That means the biofertilizer Trichoderma rhizae can increase the transportation of minerals as well as flow of water inside the plant once the plant is treated with this. And not only this, it is also able to modulate the plant genetic expression, gene expressions. Here I am showing you the real-time PCR result of genes, multiple genes such as iron utilizing gene, then this is the nitrogen utilizing gene, phosphate solubilizing gene, and further ammonia as well as sulfate utilizing genes, which are you can see the differential expression. This is control, and as compared to control, if it is increased, it is denoted in color green, and if it has been decreased, the color is changed to this. So you can see the difference is quite marked. They play, the fungi here in case of this Trichoderma rhizae plays with the genetic system of the plant so that they could tolerate the stress, and it helps the plant to tolerate the stress by modulating their expression of genes. Yeah, so this was how they were controlling the abiotic stress, how plant growth promoting fungi were controlling the abiotic stress, and now they are capable of controlling the biotic stress as well in multiple of plants. Here you can see these all four are plant pathogenic fungi. So these are pathogens. This is Sclerotium rhopsii, Rhizoctonia solani, Fusarium oxysporum, and Alternaria brassicicola. And this green thing which you are seeing, this is Trichoderma viride, Trichoderma species, which has been developed as plant growth promoting fungi. This is plant growth promoting fungus. So here you can see the growth is the plant pathogenic fungi are growing, and here you can see once the Trichoderma is applied, the growth is quite restricted. See, in all the four fungi, all the four pathogenic fungi, the plant growth promoting fungi is restricting their growth. And how they are doing so, you can see here. This one is Alternaria, and this is the mycelia of Trichoderma. You can see how it is coiling the mycelia of Alternaria, and it is directly killing it. So actually, Trichoderma viride kills the pathogenic fungi by just tangling outside them, and they are also quite well capable of producing siderophore. Now, what is siderophore? Siderophores are the iron chelating agents. Every living organism requires iron for their growth. So what does Trichoderma do? This Trichoderma, it chelates all the iron present in the environment, and thus deprives these all pathogenic fungi of iron and kills them. Here again, plant test has been performed with 4 treatment. This is the chickpea plant control. That means no treatment has been provided. And here you can see it has been treated with Trichoderma viride, and you can see a clear difference between their height and their health. And here the plant is infected with Sclerotium rofsei, this fungi. And you can see how they are getting dried and how the plant is not healthy. But once the infected plant is applied with Trichoderma viride, which is plant growth promoting fungi, the growth is again good, and that the change in leaf color, the senescence has been overcome, and the plant is now healthy. So now you can see how a simple fungi, which is plant growth promoting in nature, can ameliorate the biotic stress and protects the plant from different kind of pathogenic infection. And this is not only so, it can increase the secondary metabolite of plants. It can, say if it is tomato, it can increase the lycopene content in tomato. And after application of Trichoderma viride TP9, the stress marker, this is proline stress marker, it has also been reduced. You can see here the color is quite light, because the plants are not facing that much biotic stress that were facing in this after pathogenic infection. So this is how it is also controlling the other pathogenic fungi. One fungus is controlling the other fungus, and this is the use of plant growth promoting fungi in bioprospecting in agriculture. Not only this, they can be used for synthesis of nanoparticles. I have used Trichoderma viride, the same fungi which I was talking about, to synthesize the nanoparticles, and you can see the multiple kind of nanoparticles with different shapes and sizes have been synthesized. Here the size is smallest. You can see here the triangular one, then pentagon and hexagon, and their sheets and triangles. And you must be wondering why to synthesize these multiple kind of nanoparticles. But as you must be knowing that nanoparticles are the need of the day. They are being utilized in every field, as bioprospecting, in medicine, in drug delivery, in agriculture, in biosensing, every field. So depending upon the shape and size of nanoparticles, they can be utilized in multiple fields. So with the help of one fungus only, these different kinds have been synthesized, and this is the mechanism how they are being synthesized. So for the growth of nanoparticles, two steps are being followed. First is nucleation and second is growth. So once the nuclei is formed, the Trichoderma viride cell-free extract can synthesize multiple shapes and size of nanoparticles by modulating temperature, pH, and other culture conditions. So you can see the power of one simple fungus, that how it can help in agriculture also, how it can help in synthesis of nanoparticle also, and these nanoparticles can further be used in controlling the plant diseases. They can be used in plant disease management. Here you can see again the tomato plants have been used, and this is a diseased one. And after treatment with nanoparticles, which were synthesized from Trichoderma viride, the plants are quite healthier. The increase is quite evident in their fresh weight, dry weight, and the spore count of the pathogenic fungi has reduced significantly after application of nanoparticles, which is synthesized from Trichoderma viride. Again, the mechanistic view was also studied, that how the biogenic nanoparticles can help in controlling the plant disease, what they are doing inside the plant. So for this, the model pathosystem, as we study rat and mice in animal system, that Arabidopsis thaliana is like rat and mice of the agriculture system. So here we used Arabidopsis thaliana as model plant system, and it was infected with Alternaria brassicicola AB. You can see after infection, the growth is quite hampered and the plants are not healthy. But once they are treated with the nanoparticles synthesized by Trichoderma viride, the silver nanoparticles, their plants are growing quite well, and again their health in terms of dry weight, number of heights is quite good. And in this study, the mechanism how the nanoparticles synthesized are controlling the disease, and if they are having any effect on plant, was also studies. And this is the protein expression. This has been done. This study is the differential proteome of plant and the different treatment. And as we can see here again, that the proteome is quite modulated once they were treated with nanoparticles, and the multiple of proteins which were related with the plant defense, plant metabolism, plant ROS production were synthesized differentially. And here you can see how this is the fungal spore of Alternaria brassicicola, which is the pathogen. You can see here they are healthy pathogen. You can see here, and here you can see after treatment of particles, nanoparticles, they are dead. You can see a clear change in their structure, and they are quite dead. And what they did inside the plant, there are multiple ways of cell death, and one is autophagy, where plant utilizes the dead part for the growth and for the growth of itself, and it does not allow the fungi to feed on the dead part. So this was a mechanism which was employed in this case. You can see multiple of cellular organelles are have been accumulated inside the this autophagic vesicle, and here this is connected with chloroplast. You can see this is the chloroplast, and so this procedure was termed as chlorophagy because it was quite related with the chloroplast. It was termed as chlorophagy, and this was what happening when the plants were treated with the infected plants were treated with the biogenic nanoparticles synthesized by Trichoderma viridi. And this was another aspect of fungal bioprospecting. And these synthesized nanoparticles can not only be used in agriculture, but they have, as I told, multiple applications. So they are also used in bioremediation. Bioremediation is also very essential nowadays to protect our environment. And here we can see that this picture is all about the shape and size dependent bioremediation. As I told you in the earlier slide, that the different shapes and size of nanoparticles display different kind of effects. So here you can say that the pentagonal and hexagonal nanoparticles were the most effective one in degrading 4-nitrophenol. This peak denotes 4-nitrophenol, and here one more peak is emerging, that is of 4-aminophenol. So with the help of that pentagonal and hexagonal nanoparticles, this 4-nitrophenol has been degraded, and this 4-aminophenol has been converted into 4-aminophenol, which is following a pseudo first order reaction. And to confirm this, further HPLC was carried out, and you can see here again in case of pentagonal and hexagonal nanoparticle, the maximum this peak shows 4-aminophenol, which is highest in case of pentagonal and hexagonal nanoparticles. So this is how a single fungus or maybe different fungi can help in agriculture directly, or can be used in synthesis of nanoparticles, which is also a production of fungi only, can be utilized in bioremediation or again plant disease management. And further, fungal bioprospecting in healthcare. Apart from agriculture and bioremediation and environmental studies, these fungi can also be used in synthesis of multiple kinds of drugs in healthcare systems. So this is the study how the fungal synthesized nanoparticle was better than chemically synthesized nanoparticle in terms of antimicrobial agent. What we did, the biological nanoparticle synthesis was through fungus, and it was coated with the metabolites of that fungi, and that cumulative effect gave better antimicrobial results when applied on Pseudomonas and Staphylococcus aureus, which is quite well known for developing MDR. And how they did so? They did so by increasing the internalization inside the cell. You can see here that the nanoparticles which were coated with the metabolites of fungi, with the fungus Trichoderma viridi, were able to penetrate cells in a better capacity. And thus, as they were able to penetrate the cells, they were able to generate higher amount of ROS in all the 3 pathogenic bacteria. And so, as we can see here, that this is the nanoparticle which is being coated with the metabolites of Trichoderma viridi, and so the both nanoparticle as well as Trichoderma viridi extracts, the secondary metabolites, resulted in complete cell death of pathogenic bacteria. And whereas the chemically synthesized particles, which were having only the nanoparticle one, no synergistic effect, could not inhibit the growth of pathogenic bacteria. So this kind of nanoparticles synthesized by fungi can be used as better antimicrobial agents. Today we are hearing even now and then that MDR is being developed, that antibacterials are not effective against the bacterial pathogen. So it can be a good substitute for the current antibiotics, which can be used against MDR, multi-drug resistant pathogens as well. Yeah, so it was all about antibacterial. Now The fungal mediated nanoparticles were also better antifungal agents. So earlier, I don't know much about that people were knowing about the antifungal agent, but after that COVID-19, multiple cases of mucormycosis were reported. So that mucormycosis is also a fungal disease. So fungal mediated that fungal bioprospecting comes here also, and the nanoparticles synthesized by trichoderma were able to control different kind of fungal pathogen in a better manner. You can see this is the biologically synthesized nanoparticle, and the red color shows the death of Alternaria fungi, which is pathogenic in nature. And here we can see after treatment of that fungal nanoparticle, the death is quite evident. Again, here we can see that this is the healthy mycelia of Alternaria, the pathogenic fungi, and this shows the treatment of chemically synthesized nanoparticle. And here you can see, yes, the destruction is there, but it is not quite prominent. And here in these two pictures, we can see that after application of fungal mediated nanoparticles, the structure of this pathogenic fungi has completely disoriented. It is completely destroyed. So here also it is quite evident that the fungal mediated nanoparticles coated with the metabolites of trichoderma are more capable of killing the bacterial as well as fungal pathogens, which can be used in hospital settings and other medical settings. And now, again, we come on the role of direct fungi. So, as I told earlier, that fungi are prolific producer of different kind of secondary metabolites. They can produce n number of metabolites. And in my lab also, the Taxol, which was earlier produced by Taxus baccata plant only, has been reported to be produced from a number of endophytic fungi. And here I am showing you fungal extract. This is a TPEE, that means this is Telleromyces purpurogenes is the name of fungus, and this has been isolated in ethyl extract. That is why it is known as TPEE, and it is showing a very good effect on multiple of cancer cell lines. You can see HeLa, cervical cancer, breast cancer, and pancreatic cancer, lung cancer, and brain cancer cell lines. It is showing a very good effect. The death rate in HeLa, you can see it is quite good. Even on 50 microgram per ml, the death is approaching almost to 50% in cancer cells. And the mechanism employed was it changed the mitochondrial potential, as well as it increased the reactive oxygen species, and it further led to apoptosis. Apoptosis is one of the most general mechanism through which cancer cells are being killed by the chemotherapeutic agents. And this is one pictorial representation. This is the confocal image of acridine orange and propadium iodide staining. Here you can see this is the control and control one. That means the healthy cells of HeLa, the healthy cancerous cells. And here after the application of TPEE, you can see the blebbing of cells here all. Blebbing is the preliminary situation of apoptosis. After that, cells eventually turn into the apoptotic body and dies. And this orange color indicates the late apoptosis, which ultimately leads to the death of cells after application of fungal extract. So this was the story of one extract, and here we can see that after the next step is purification of extract. After purification of extract, multiple compounds can be isolated and identified, which can further be used in as anti-cancerous agent. Here I am showing you the example of one Chaetomium globosum. This was one endophyte. This is one endophyte which is isolated from one green algae of Konkan coast. And for the first time in literature, the chrysine, which was generally isolated from honey, has been isolated from one fungal source which is marine in nature. So you can see the potential of one fungus, that it can produce numerous compounds. It can substitute the plants, the animals, the other microbes for synthesis of fungi, or for synthesis of natural products. And here how it was identified from FTIR and CMS. And here again we can see that it is quite potent in killing the cells, and it arrested the cells in G2M phase, produced ROS, and it also induced the loss of mitochondrial membrane potential, ultimately leading to cell death of the MCF7 cells, that is breast cancer cells. And so this is how the Fungi are quite valuable things which can be used in agriculture, in nanoparticle synthesis, in bioremediation, and in healthcare. But there are certain hurdles and which are associated, which definitely can be overcome. So the first one is legal and regulatory complications. So yeah, one has to go through a number of things before one drug reaches into the market or one biofertilizer reaches into the market. There are a number of clinical trials and all that have to be taken care of for the so that the fungal bioprospecting can reach to the market. And one most important thing is biopiracy. Biopiracy, that means, as I told earlier, that bioprospecting is generally associated with the indigenous people, tribal people, the people who are native to that place. But generally, what big pharma company does, they do not give them their credit and just patent or something like that on their name. You must have heard about the basmati and that neem case, where the foreign companies have patented the basmati and neem. Though we all know that their potential benefit is quite evident in India, but CSIR fought and brought that product back to the India. So this should be avoided in fungal bioprospecting, so that everybody should get the credit and the benefit of bioprospecting. And yes, before any of the product reaches to the market, that has to be made clarified that it does not harm our native microflora, whether it is the plant system, the native plant microflora, or our gut microflora. It should not impact that negatively. And costing and sustained delivery, yes, of course, these are the most important things to be looked after. Costing, the cost should be in range of a common man, so that that can be further used in multiple purposes. And sustained delivery, that means most of the natural products, say secondary metabolites of fungi, are not soluble at physiological pH. They are generally hydrophobic. So for this, they can be semi-synthesized chemically, or nano delivery systems can be used for their delivery. And these are quite researched upon in the research community, and mass awareness is also needed for the further growth of fungal bioprospecting, which can be used in agriculture and healthcare. As I give you one example of NBRI, they are continuously in touch with the farmers of UP, and they are giving the slants of inoculum of sample of Trichoderma viridi as plant growth promoting fungi to farmers, so that their growth can be the growth of plant, the growth of, say, cereals, or whatever they are cropping, that can be increased. So that kind of interaction is also necessary. So yeah, this is all about the fungal bioprospecting, and I would like to thank everyone in my lab, in my earlier lab, in my today's lab, as well as Parul University, and especially to my CK sir, who gave me this opportunity to interact with you all and present my work here. Thank you all.
Thank you, ma'am. Thank you very much for enlightening us and providing a deep understanding of fungi and summarizing its various applications in the field of microbiology and sustainable agriculture. Friends, all of us know that fungi bioprospecting has a greater role in sustainable agriculture, and it has explored huge potential of the many fungi to the human lives. And the latest developments and scientific advances in the study of biodiversity of fungi or extremophilic fungi and fungal secondary metabolites and enzymes has a greater significance. So thank you once again, ma'am. Now I would like to request Honorable Dean sir to have chairman's remarks on the present session.
Thanks, Dr. Nitin. Today I am very much happy to see one of my students from the ISC Bangalore, Dr. Madhuri, has addressed the gathering. And addressed as the CK sir, which is the pattern for me among the students who have been taught by myself, to kind of a different movement which I can realize, which I can feel. And whenever the students move, and if the students move ahead of the teacher, that looks very kind of, pleasant for a teacher. I can recall the days when I taught Madhuri in the classroom also, and I'm glad that she has quoted so many things which the nowadays the advanced science is doing. And I'm also glad that my research scholars, they are working on the nanoparticle synthesis. And today, it is very much valuable for the science graduates that the multiple and ample opportunities are available to work on such aspects. After going through such kind of the knowledge or thought-provoking lectures, we always think how much we have to do in the science.
Yes. Mm.
Still, we have to harness something which is not understood till today. So, Madhuri, no doubt, I have been your teacher, but I have a kind of query as you are working, and I am glad on that.
Yes. Yes, sir.
Suppose you have utilized the nanoparticles for the synthesis of the textile, as you have quoted, that can be utilized for the textile synthesis. And as you have utilized the nanoparticles which have been coated by the secondary metabolites regarding the cell senescence.
Mm-hmm.
So generally, we use some kind of polymers
Yes.
for that purpose also, like as the PLJ, we use of that. How you compare that with this kind of exercise what you have done in that case?
Yes, sir. So initially, my work was based on the metal nanoparticles. So I thought to how we can increase the potential of metal nanoparticles, as silver nanoparticle is being used in many of the washing products, many of the toothpaste as well. So I thought how we can increase the efficacy of one simple silver nanoparticles. So I coated them with trichoderma viridi extract. And now, further in my current research, I am working with the secondary metabolites. So, sir, your point is quite true that here we cannot use that kind of, those kind of tactics. Those are different one. And here, in my current research, I am using chitinase and PLG as polymers to coat the secondary metabolites for the sustained delivery. As I told that these are hydrophobic in nature, so for the sustained delivery, these are being used.
Naturally, actually, my students have done the work on that PLG basically, and we have utilized that. From our esteemed Paro University, I want to put that at our Faculty of Agriculture, we are offering the different kind of the courses. That is the BSc Agriculture, BTech Dairy Technology, BTech Agriculture Engineering, the Animal Husbandry, and the Food Technology. So, since you are the scientist there, if any kind of the possibilities for a kind of tie-up or kind of mutual benefit with the ISC Bangalore and the Paro University, I think that will be good for our students also. So it is my request, being a dean, and it is my urge, being a teacher of yours, please find out some possibility. If
Sure. Sure, sir. I will try to find out the possibility because I'm also working on the polymers and the secondary metabolites. So I will try to find out whether we can work together.
Indeed. So, Nitinji.
Yes, sir. Thank you, sir, for your Atlantic speech and interaction. As sir, already we have discussed so many ideas regarding the interaction with the eminent personalities as well as the collaboration with them. Hope ma'am's guidance will surely enlighten our students. And if furthermore PU collaborates with ISC, that will be a big achievement for students as well as the institute. Hope so it would be true in the near future. As ma'am, I have a question. How these fungal endophytes can be a source of fuel in the or biofuels in the near future?
Yeah, so fungal endophytes, as I told, that these are quite potent in generating those kind of secondary metabolites which are generally present in their host. So multiple plants such as jatropha is being used for production of biodiesel or something. So we can isolate the endophytic fungi from those kind of plants, and then maybe we can search from the possibilities of that biodiesel and biofuel production, as well as these can be used in production of biosurfactants, which can enhance the oil recovery. So in this aspect also they can be used.
Yes, thank you, ma'am. Actually, in such type of geopolitical issues currently going worldwide, biofuels could be an alternative.
That is
Hope
true.
so. We will make it in the upcoming decade nationwide.
Yeah, I also hope.
I am very much thankful to Honorable Dean Sir for providing me an opportunity to moderate this webinar. And this is one of the platform has been provided by our dean sir to all the faculty members to strengthen themselves, and we get opportunity to interact with such eminent scientists personalities, and that's the greatness of our dean sir to provide us platforms. So now with these words, now I'd like to request dean sir to share his thoughts in the end of the session.
Thank you very much. Thank you very much. I think Madhuri is very much perfect in that. And,
Thank you very much, sir. Thank you very much for your all guidance and everything.
thank you very much, Madhuri.
Thank you, sir. Thank you for giving me this opportunity. Thank you,
Thank you
sir.
from the PU family.
Thank you, sir.
Thank you.
All the
Okay.
best.
With the permission of chairman, I declare the session is over. Thank
Thank
you,
you.
ma'am.
Thank you.
Thank you.